Hello, everyone. My name is Xander, and I am a student at the University of Iowa, and I will be helping you guys go through this first worksheet for General Microbiology 2157 with Dr. Regina McGrain and Dr. Jana Radin. So this is the first Chapter 1 worksheet. They also have videos as well, but I'm going to kind of explain it in a way that I guess a student point of view, and if this helps you guys more, great. Let me know what else you guys want me to do video wise. I can do a lot of different classes, so put anything in the comments that you'd like to see school wise and I can help with. So without further ado, let's get into it. So we have the first question. It's a Venn diagram comparing eukaryotes and prokaryotes. So first thing we have is DNA. So DNA is present in all cells, anything that has part of cellular life. And then for chromosomes, definitely know the differences between these. So eukaryotes have linear and prokaryotes have circular chromosomes. All right. And then ribosomes are present in all cells. And these are kind of in the cytoplasm of the cells, and this helps with translating the mRNA into proteins, making, that process, which you'll learn about more. Also know this differentiation, so eukaryotes are 80S ribosomes, and prokaryotes are 70S. For cytoplasm, all cells have cytoplasm present. And then plasma membrane in all cells. And if you remember, it's pretty much like this. And I'm kind of lazy. There's technically a better way to do it like this. So, but essentially, these are the hydrophilic heads, so they like water on the outside, and then these are the hydrophobic tails that are kind of that lipid soluble area that don't really like water, and they also only let certain things go through. It's selectively permeable. So then membrane bound organelles, that's only for eukaryotes. So for example, mitochondria, chloroplasts, etc. Prokaryotes, technically, we would say for now, don't have that. Replicate by binary fission, that would be prokaryotes, which you'll learn kind of more about that process later in the class for microbes. And then eukaryotes having mitosis and or sexual reproduction. That's pretty simple, self-explanatory, so try to kind of have this to the T, just be able to know this very quickly. Secondly, when looking through despite looking very similar microscopically, what was the first evidence that bacteria and archaea should be classified as unique domains? So this is kind of the concept with our guy Carl Woos, or however you say his name, and he was analyzing rRNA sequences, and he was noticing these kind of unique rRNA sequences that were different from bacteria and eukaryotes. So he kind of created or named this third form of life called eukarya, and it's right here in my notes. I'm going to go flipping back and forth through my old notes to kind of show you exactly where I'm getting it from. So first, to identify a third form of life by comparing ribosomal RNA sequences. So it's exactly what I wrote. And it was the rRNA that he discovered from methanogens, which are pretty much those first archaea. And if I go here, these were the ones that would produce methane as a waste. There's also the other types of archaea, but for this worksheet, that's not really relevant right now. And also, as a quick thing again, the all cells, the part we just did, have DNA, ribosomes, plasma membrane, cytoplasm. And then with comparing prokaryotes to eukaryotes, here's the ribosome differences. And you can think of ADS as like bigger, and because eukaryotes are typically multicellular and like bigger organisms. Larger genome, linear chromosomes. Even though this is kind of an X, their linearity is they become very condensed, and that's kind of why they form that X shape. What else? And oh yeah, so the Carl Lewis, we kind of already figured that out. That's all you really need to know, is these are the main things from my notes, and what I kind of wrote here. These are the big concepts. And then for 4 reasons to study microbes. So there's kind of a lot of different reasons here in my notes. Let's see. There's the aspect of food, so can cause food spoilage, foodborne illnesses, food poisoning. It can also be used, actually, the fermentation process that microbes do can be used for making cheese, milks, yogurts, etc., and like acidic foods. And it'll contribute to flavor as well as actually preserving the foods. So microbes are not all bad, they can be used for a lot of beneficial things. On top of that, there's also the human microbiome, which is very important for our body to help keep our health intact, helps maintain good health, and helps us kind of absorb our nutrients and whatnot. It also kind of outcompetes pathogenic microbes to make sure that they don't pretty much overtake our body and cause too much damage. There's also, what else is there? Ecology. So yeah, it can benefit plants and animals as well as causing disease. So as long as you kind of have 4 things, you should probably know most of the things that I was talking about, but if you can kind of be able to easily know the importance of microbes, then you should be okay. So back to another Venn diagram, including bacteria, archaea, eukaryotes, and prokaryotes, so a little bit more. We have the same concept for all cells, so DNA, ribosomes, plasma membrane, and cytoplasm. And then eukaryotes, ADS, ribosomes, prokaryotes, 70S ribosomes, eukaryotes, linear chromosome, prokaryotes, circular chromosome. Yep. Membrane-bound organelles, eukaryotes again. Replicate by binary fission, prokaryotes. And they'll kind of go deeper into this, so I'm just kind of helping you with the thought process of this worksheet. Replicate by mitosis and sexual reproduction for eukaryotes. So when you think of eukaryote, you can also think of us, humans. We have mitosis for our cells as well as sexual reproduction. What else we have? So we have membrane-bound organelles, right? So that includes mitochondria, chloroplasts, which these are kind of part of that endosymbiotic theory, which I would definitely kind of understand that concept as well. Anything in the learning objectives as well. So knowing these learning objectives, definitely study and practice this. So these are the main things they're going to be testing in the class, I promise. I didn't trust it at first, but it is definitely true. Peptidoglycan. So this, very important. Bacteria only, peptidoglycan. Cell wall. Only bacteria have that, okay? Archaea. S layer, and pseudomurine cell wall. Eukaryotes, they don't really have cell wall. Bacteria, peptidoglycan, remember that. This will be important when you learn about gram staining with gram positive, gram negative, which should be a little bit later. And then that archaea are S layer and pseudomurine. And that should be it for that aspect. Okay. So if two organisms are referred to as protists, is it appropriate to conclude that the organisms are close relatives? Why or why not? So, no. This is, I will pull up in my notes to show you. Where is it? Yes, so right here. So eukarya are single-celled or multicellular, right? The idea with protists is they are single-celled eukaryotic microbes that are not fungi, animals. They're pretty much not exactly like the rest of the eukarya. So we have protozoa, which are animal-like protists. They lack the cell wall like animals. And then there's algae, which are like the photosynthetic kinds. And the reason for this question that it's not appropriate to consider them relatives is because it's not monophyletic, which essentially means that on this tree, this phylogenetic tree, let's say, I already kind of circled it, but this can be related to like something over here. So like, for example, these things are all related, right? These three, or let's say theoretically, but like you can have a protist that's like here and like one that's up here, and they're very similar, but they're obviously not in the same group because this one's down here and this one's up here. So protists are not really classified through a phylogenetic tree because they don't follow that same aspect of other things. They are typically polyphyletic. So that's kind of exactly pretty much what it says in the worksheet. 3 characteristics of viruses that make them distinct from cells. So, I mean, there's kind of a lot. Acellular, I mean, they lack ribosomes, so they lack kind of a lot of machinery that cells have. They're also obligate parasites, so that require host cells to be able to replicate. So they need to pretty much, they need to get into a host cell to kind of do its functions, right? So that's kind of a difference, as well as they cannot create or catabolize things, break things down to create their own ATP. So that's kind of an important factor as well. They include RNA or DNA. It's not necessarily both. And one kind of distinct thing as well is they have this structure called a capsid, which is the protein coat. So it's made of proteins and it surrounds the genetic material of a virus. So if you see right here, I can kind of draw it out bigger. So this is like a typical bacteriophage, right? It has its protein coat with the genetic material inside, right? So this is DNA or RNA, whatever. And then it'll kind of look like this, right? With its like little legs out, just like this image. And then they sometimes have envelopes as well, which is made actually from the host cell. You don't really need to know exactly that right now. But just know some differences and to be able to distinguish like what a virus is from a cell. Genus name and the specific epithet. So for this one, the first word of these is always going to be the genus. So we can just circle these first 4. And then specific epithet, we put a star next to it, so right, okay. And then if you notice, these all have capital letter for the first word and lowercase for the second one, and they're all in italics like this, right? So that's the 3 formatting requirements that actually kind of answers the next question. So I'm just kind of numbering this. It doesn't matter how it's numbered. So italics and then first word is capital, and then second word lowercase. So just like up here. And this will become like second nature after you kind of learn about the different microbes, just kind of throughout your class and throughout the semester. So now we're kind of down to identifying people. They do want you to know some people, so definitely kind of know the ones from this. The first person that's kind of credited with naming cells and describing the first observations of fruiting structures of molds, this is going to be Robert Hooke. So if we go to my notes up here, yep. And this was from the corkscrew experiment, which she will explain in lecture, with the kind of single lens compound microscope. So that's pretty self-explanatory. It's just knowing the name and trying to remember that. Then we have credited with recording the first observations of bacteria. So this guy actually called them animalcules at first, Antony van Leeuwenhoek, however you want to call him. He describes what we now know as bacteria and protists. So he called them animalcules. So this is Antony. Sorry, excuse me. Give me 1 second. All right, sorry about that. So, on to number 10. This guy kind of did a lot. So he disproved spontaneous generation, right? That concept that like microbes just come from nothing, which when we think about it now, that obviously doesn't really make a lot of sense. So that was Louis Pasteur, kind of that final guy with the swan neck flask experiment and trapping the microbes with the broth and the sterilization and whatnot. He found out about disproving that, right? And then he also described that immunity to chicken cholera down here, Louis Pasteur, so inoculating them, and having that kind of attenuation concept of developing vaccines and pretty much having the pathogens losing their ability to cause disease, which she'll explain further. You don't really need to know all that because it's just answering who it was, And then we have identified the cause of fermentation. So he also discovered, I'm trying to think if I had, here, that microbes carry out fermentation. This is related to like the yeast with the alcohol concept that was explained. So he also was part of that, as well as developing pasteurization, heating things up to like kill microbes and whatnot. And he also developed the rabies vaccine. So this is Louis Pasteur. And I mean, his name kind of gives it away. All right, and then describing similarities between Pasteur's fermentation experiment and Koch's postulate. So these both kind of were designed to target, or I guess connect a specific microorganism to a specific process. This question, I don't harp on this too much. I feel like when I was doing this originally, when studying for the class, I feel like I was kind of reading how they explained it, and I was like, I don't know, I feel like it's kind of a reach to try to find similarities. So just kind of understand the fermentation experiment and like the postulate with Koch about like these 4 steps to try to kind of have that in your head. I don't think you really necessarily need to memorize it, but I would definitely say to kind of know what the steps are generally. So why might his postulate sometimes be difficult to fulfill? So first of all, like, suspected microbes must be isolated and grown in a pure culture. Okay, well, that's kind of a problem because only about 1% of microbes are culturable. Right, and I have that in my notes in this bottom corner. This is when you guys are learning about how to study microbes and like pure cultures. This is kind of a big highlighted point. Then, what else? Are there any microbes to be discussed that cannot be grown in pure cultures? So yeah, I don't actually think, did they answer this? But I would say for this, viruses, right? They need a host cell, so they can't just grow by themselves, right? There's also other aspects, like many different multiple pathogens can cause same symptoms. And yeah, that kind of just goes against like his postulate if you kind of read into it. Those are kind of the main things I would say. Then the last two, we have developed a vaccine for smallpox using the cowpox. So this is Edward Jenner. This is the milkmaid experiment. Pretty much inoculating or putting cowpox and smallpox into kids and seeing how the kids were not affected by smallpox. So pretty strange experiment. Obviously, things were different and ethical things were different, but yeah, that was through Edward Jenner. And then lastly, demonstrating the metabolic diversity of the environment microbes. This is Sergei Winogradsky. This is involved with the Winogradsky column, which you might have done in high school. Kind of having a bunch of different microbial environments and different layers of growth, and it kind of just helps you learn about the diversity of microbes, so it's kind of an interesting aspect. That is it for this worksheet. If you guys have any questions or want me to do any other material, let me know. And thank you very much. Have a good one. Bye-bye.